The concept of heredity, which refers to the transmission of traits from parents to offspring, has existed for as long as people have existed. Farmers and livestock breeders have long understood that offspring usually resemble their parents. However, despite intensive research, the mechanisms of heredity have remained a mystery for millennia until recent times. People used to hold a wide range of beliefs about heredity. The majority of these were founded on observation and hypothesis rather than an understanding of how genetics worked. Here are some examples. It was once widely held that offspring possessed a blend or mix of their parents' characteristics. If one parent is short and the other is tall, the children will be of average height. However, this theory was unable to explain how traits that had been lost in a previous generation could suddenly reappear in succeeding generations. Preformationism held that all organisms formed at the same time, and that each embryo or egg contained a complete, miniature organism, known as a homunculus, that grew larger as the organism developed. This theory is unable to explain how traits inherited from both parents may manifest in offspring. According to the Lamarckism theory, an organism can change in response to its environment during its lifetime, and these changes are then passed on to the organism's progeny. Jean-Baptiste Lamarck proposed this idea. Although not entirely accurate, the example of the giraffe's neck growing across generations in order to reach higher vegetation is commonly used to demonstrate this. The pangenesis theory, proposed by Charles Darwin, states that every part of the organism produces particles known as gemmules. These are then accumulated in sperm and eggs and passed on to the next generation. This hypothesis attempted to explain how changes that an organism acquires over the course of its life can be passed down to its descendants. The first major breakthrough in our understanding of heredity occurred around the middle of the 19th century, thanks to an unusual source, a monastery in what is now the Czech Republic. Between 1856 and 1863, an Augustinian friar named Gregor Mendel was responsible for a comprehensive set of studies on pea plants. Mendel painstakingly crossed plants with distinct characteristics. He then observed the progeny's characteristics over many generations. He was able to deduce two fundamental principles of inheritance from this single piece of evidence: the law of segregation and the law of independent assortment. However, the modern era of Mendelian genetics did not begin until 1900, when Gregor Mendel's work was discovered. At the turn of the 20th century, scientists began to associate the physical manifestation of heredity, traits, with their location within the cell. The chromosome theory of inheritance was proposed independently by Walter Sutton and Theodore Boveri in 1902. They proposed that the behavior of chromosomes during meiosis could explain Mendel's laws. It was recognized prior to the 1950s that chromosomes carried genetic information. However, the exact molecule responsible for this was unknown. Oswald Avery, Colin MacLeod, and Macklin McCarty conducted a series of tests in 1944 to demonstrate that DNA is the substance responsible for bacterial transformation. This established that DNA serves as genetic material. In 1952, Alfred Hershey and Martha Chase conducted studies using bacteriophages, which are viruses that infect bacteria. These investigations corroborated the previous conclusion. The discovery of the structure of DNA should be credited to a group of researchers who collaborated to discover the now famous double helix. The double helix concept, devised by James Watson and Francis Crick in the 1950s, sparked the revolution in genetics and molecular biology. Their discovery was a huge accomplishment, but it was built on the work of other scientists who contributed significantly to the scientific understanding of DNA. Rosalind Franklin, who specialized in X-ray crystallography, was one such researcher. Photograph 51 is a photograph depicting a blurry X in the center of a field of black and white splotches. This X-ray diffraction image of DNA was crucial to understanding how the double helix structure of DNA is organized. The recognition given to Watson and Crick for their contributions to the discovery of the structure of DNA obscured the important role that Franklin played in the discovery. Furthermore, Franklin died in 1958 as a result of ovarian cancer. She passed away 4 years before Watson, Crick, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for their work on DNA.